Next, some basic principles of design of experiment. There are basically three principles. So first one, randomization. Second one, replication. Third one, local control. Randomization. So in an experiment, if the testing samples or the experimental participants are selected randomly from the population, then it is called randomization. So in a class there are 30 students. If you want to select one student to do some leading, that is some work, class related work, right? So you have to call any person from that 30. You have to select randomly from that. That is called randomization. So you can choose any of one from the number of students, that is total number of students, that is from the 30 students you can choose any number of one, right, from that, right? That is called randomization. In that case, so all are getting equal chance to be selected, right? That is called randomization. So randomization is a technique in which two or more treatments are applied to different group of the sample. The treatments to be applied to any group being decided by random sampling techniques. That is, suppose if you are having 3-4 treatments, so if you are we are going to that paddy field, if there are 10 paddy field, then you are having 3 fertilizers, right? And the type of paddy, so there are different type of paddies, right? Seeds, paddy seeds, right? So there are 3 or 4 types of paddy seeds. Then you have to put it in the first field. So you have to choose the field randomly. Out of 10 you can choose any field. Out of 4 paddy seeds you can choose any one of them randomly. Then the fertilizers, out of 3 fertilizers you can choose any one fertilizer in that particular field. That is called randomization, right? Suppose there are 60 persons participated in a study of comparing two energy drinks, drink 1, drink 2, right? Each person could be selected randomly to have either drink 1 or drink 2. So there are 60 persons, right? Participants. So randomly we are selecting group 1, that is 30 persons from out of 60, randomly, right? We are choosing. Similarly, we are choosing group 2, which contains 30 persons, right? Randomly. So treatment group 1, they are taking the drink 1, right? And treatment group 2, they are taking the drink 2, right? We are checking in that. This is called randomization, right? Next, replication. The process of repeating an experiment or set of treatment is known as replication. So you can repeat the same test again and again. So each time you will get different outputs sometimes, right? It is not in the physics or chemistry. Just if you are doing the practical experimental, right, paddy field, right, whatever it is, if you are getting if you are giving same treatment to same field, so but yield will be different, right? It will not be same. That is what replication is, repeating the treatments or the total experiment, right? To know the average height and weight of a person, for example, in a city, if we selected a person from the total population of the city and measure his or her height and weight, from this one sample we cannot conclude that the average height and weight of the total population of city. If you want to know height and weight of the person, right, average height and weight of the person in your city or in your street, you cannot conclude that if you are choosing any person from your street and measuring his height and weight, from that all the person in the seat will be the in the same height and weight. That is not the correct output, right? So but the replication of this observation through random sampling of 100 more individuals from the population of the same city. Instead of that, you can choose or 50 to 75% from your street, right? You can measure the height and weight of each persons and take the average. Then you can get the almost maximum absolute maximum nearby the absolute height and weight, average height and weight of the persons living in your street. So we need to replication, so repeating the process for the different persons, the same measuring the height and weight for first person, second person, repeating the, this is called replication in this case, right? So we'll give a conclusion about weight, height and their interrelation, right, in the city of population at large. Next, local control, the process of reducing the experimental error. So to avoid the experimental error, so the process we are doing, process arranging the experimental units in the blocks. So we are changing the units in that blocks. So experimental units, first one, second one, third one, we are reversing third one to first one, and we are rearranging to control the error, right, that output error, right, that is called local control. It is the process of reducing the experimental error by arranging relatively the heterogeneous experimental units. So suppose in that block, if you are having there is 2 or 3 fields or different soil type, just from the almost other, right, 7 are in the almost same type, and out of that 3 is some slight difference are there in the soil, then you have to keep separately that 3, right? So then the output will be almost same for the 7, right? That is called local control. Heterogeneous means that is the so if you are having different type of soils in one block, that is heterogeneous, right? Homogeneous is same, all are almost same. Into homogeneous, right? Example, for instance, an experimental field is heterogeneous with respect to soil fertility, then the field can be divided into smaller blocks such that the plots within each block tends to be more homogeneous. This kind of homogeneity of plots, that is experimental units, ensure an unbiased comparison of treatment, right? This type of local control to achieve homogeneity of experimental units, right, will not only increase the accuracy of experiment but also help in arriving at valid conclusion. That is what. Next, aim of the design of experiment. The main objective of design of experiment is to control the irrelevant variables other than experimental variables. So if you want to check the output of a paddy from a paddy field, so the treatment should be variable, that is variable. So soil nature, fertilizer, rainfall, then other climate, everything are involved. These are all the treatment. In that, if you are putting unnecessary variables, so not related to paddy, then that is called irrelevant variables. Suppose instead of paddy, if you are having some plant, sugar cane, whatever it is, that is completely different, irrelevant to that experiment, right? So to avoid those things, right, so that the result could be entrust only to the experimental variable. So you can argue differently. Suppose if you want to get the output of a particular fertilizer for the particular paddy seed, right, so you have to put the particular paddy seed and particular you have to use particular paddy seed and particular fertilizers in that field, right? So the fertilizer of other type, it is irrelevant to that, right? You can avoid those things, that is called, right? Thanks.